So for this final, it was on SARS-CoV2 testing. So background information. It originated in Wuhan, China. A virus is any group of complex biochemical entities consisting of genetic material wrapped in protein. Viruses can reproduce within living host cells only. So we test the RNA expression levels, and SARS-CoV2 is a beta coronavirus. There's many coronaviruses with alpha and beta cells, but this one is a newer version, newly discovered version of SARS-CoV. So COVID-19 can spread through droplets from the nose or mouth. These droplets can be spread by coughing, sneezing, or speaking. Once a droplet from somebody with COVID-19 is inhaled by somebody without the virus, they can become infected. And these droplets can stay on surfaces for hours, depending on what surface it is. Like plastic can be 3 hours, cardboard can be 24 hours. It just depends. So it's important to wash your hands when you touch a surface, wear a mask. Just be cautious, wear gloves. So first, as of Sunday, May 8th, the United States has 1,122,486 confirmed cases and 65,735 total deaths. I'm from Auburn, Massachusetts, which is next to Worcester, Massachusetts. It's the second biggest city in Massachusetts, so I'm part of Worcester County. There are 5,787 confirmed cases, 278 total deaths as of Sunday, May 8th. We have 2 major hospitals there and a field hospital. So RT-PCR background information. A PCR is a polymerase chain reaction that is used to amplify small parts of DNA. RT-PCR is a reverse transcriptase polymerase chain reaction that measures RNA expression levels. RT-PCR is used to test for SARS-CoV2 because it detects nucleic acid associated with SARS-CoV2 within upper and lower respiratory specimens. And this is where they use like a swab to collect the specimens. A positive result means there is a presence of SARS-CoV2 RNA in the patient. This result does not mean the patient doesn't have a different virus, bacterial infection, or a co-infection. So it doesn't mean that they definitely have it, but it means they could, and they could also have a different virus. ELISA background information. An antibody-antigen complex is formed when a patient's SARS-CoV2 antibodies, which is IgG, and a coded RBD protein combine, followed by a secondary anti-human IgG-HRP antibody. The antibody-antigen complex turns yellow for positive results when sigma-aldrich sigma-fast OPD substrate is added. The shade of orange from the addition of the stop codon determines the amount of SARS-CoV2 immunoglobulin G antibodies present. The presumptively positive results have an antibody value greater than 0.15. The test is taken further with a spike protein ELISA. Both RBD antibody ELISA and spike protein ELISA must be positive to have an overall test result. So if the first test, the antibody test, isn't positive, they don't even do the second spike protein part. A positive result for a person with SARS-CoV2 would mean they currently have COVID-19. So the barbecuers. A group of friends decided to get together to have a barbecue even though they were supposed to be partaking in quarantine. Purpose of quarantine is to limit the spread of viruses such as COVID-19. One friend, Johnny, developed symptoms of COVID-19 a week after the barbecue, followed by another friend, Maura. The 6 friends decided to get their blood drawn for an antibody titer to get tested using RT-PCR and ELISA. So every person must get tested if they have symptoms, but they don't, using RT-PCR and ELISA. they could be carrying the virus without symptoms. A lot of people are second guessing. They just don't really understand that you could have it and not be showing symptoms. And that's why there's the wait period of 10 to 14 days. So hypothesis, if the it's supposed to be RT-PCR, and the ELISA test results come back positive and the patient has SARS-CoV-2 infection or has been infected previously. The RT-PCR results. So there were 10 different tests to show the results. So figure 1, sample showing results of RT-PCR test to measure RNA expression levels and nucleic acid from SARS-CoV-2. Well A, which is the blank, well B demonstrates all possible results. Well C shows a positive result for RT-PCR test, and well D shows a negative result. So you can see positive control, there's one line, negative control, there's none. Maura, Johnny, and Roland all show positive results, so E, F, and J, which means they show a presence of SARS-CoV-2 RNA. David and Patrick show negative results. David, Patrick, and Alexis actually show negative results, G, H, and I, meaning they do not have the nucleic acid in their body that is associated with SARS-CoV-2. ELISA results. So this is figure 2, a titer of samples to test for human IgG antibodies in individuals with prior suspected infection of SARS-CoV-2. To be confirmed positive for IgG antibody, the patient must test positive for both RBD screen and spike protein. Well, 1 is a positive control, so it indicates a positive result for SARS-CoV-2 IgG antibodies. Maura, David, Patrick, and Roland tested positive, so 3, 5, 6, and 8. Well, 2 indicates what a negative result would be, meaning there's no specific antibodies in the patient relating to SARS-CoV-2 in the RBD ELISA. So Johnny and Alexis tested negative, 4 and 7. Figure 3, this is what I showed you on Excel. So it's a comparison between Maura, Johnny, David, Patrick, Alexis, and Roland using the mean of 3 antibody titer samples, results of SARS-CoV-2 testing. Roland had the greatest mean, and he's on the far right. Alexis had the lowest mean and standard deviation. She's next to Roland. And Patrick had the largest standard deviation. So he had the largest difference between all 3 of his tests. Conclusion, Maura, Johnny, and Roland currently have SARS-CoV-2 infection. According to the RT-PCR results, they show presence of SARS-CoV-2 RNA. David and Patrick previously had SARS-CoV-2 infection but are not currently infected. They carry the antibody but do not have a presence of SARS-CoV-2 RNA. And Alexis has not had a SARS-CoV-2 infection at all. She received negative results from both the RT-PCR test and the ELISA. And here are my resources, my references.